Assalamu alaikum. I hope you're all doing fine. I'm Dr. Muhammad Ali Rabbani, and today we are going to discuss the development of the eye. Okay, by the end of this session, we will describe the development of optic cup. Where does it come from? How does it differentiate? We will relate the differentiation of optic cup into definitive structures. We will correlate common congenital malformations of eye and describe the development of various layers of the eyeball. Before we jump into the lecture, let us briefly review the structure of the eye, and this is the final structure that we are going to end up with. So if we see an eyeball, it is consisting of three layers: the outer fibrous layer consisting of this whole sclera, the fiber, and then cornea. So cornea makes up 1/6 of the circle, and the rest 5/6 is by sclera, and it is a tough fibrous layer that protects the eye and give attachment site for the muscles. Cornea is transparent. It's in front of the eyeball. It allows the light to enter the eye and refracts it as it does so. Then the second layer is the vascular layer, and it surrounds all of the eye. In the back, this is called choroid, and here it is specialized to form ciliary body, and in front of the lens, this is specialized into iris. Then in the center there is the retinal layer. For most cases, the retinal layer is neural in nature. It's sensitive to light, and here is where the image is perceived. And then in front of the retina, this layer does continue behind the ciliary body and iris, although non-neural, but it does continue behind the ciliary body and iris. Then we have the lens behind the iris, and that leaves us with the two parts of the eye in the cavity. There is the vitreous humor, the vitreous chamber, the jelly-like area, and in front there is aqueous chamber. Aqueous chamber is further divided into anterior and posterior parts, anterior chamber and posterior chamber, in front of and behind of iris. So these are the structures that we're going to discuss, and we're going to uh, end up here, inshallah. So at the start, let's fall back to general anatomy or general embryology. So when we will start with the development of eye, we are at this stage, roughly the 22nd stage, where the neural fold is occurring. There is ectoderm. Ectoderm is being divided into two parts: the part that remains in the skin, the surface ectoderm, and the part that becomes a part of the neural tube, neuroectoderm. Then there is mesoderm, there is endoderm, and there is neural crest cell between them. So as the neural tube is closing, still not closed, somewhere here will be the part where eye field will be generated. And what were the neural crest cells? Those were the cells of the surface ectoderm. Those were the cells of the ectoderm that were at the margins of the neural fold, that were at the margin between the surface ectoderm and neural ectoderm. And these are the cells that are going to be playing important role in the development of the eye. So when we will talk about the sources of the development of eye, we will be talking about neuroectoderm, that will be the neural tube; surface ectoderm, that will be the skin; mesoderm and neural crest cell. These two parts, those will be from both neural crest cells and mesoderm. They will be making a jelly-like area that will be making a undifferentiating mass of tissue called mesenchyme, and from that the other structures will be formed. So let us start our lecture from the beginning, that is from development of optic cup and optic vesicle. The story of the eye begins at the day 22, at the beginning of the 4th week, at the end of the 3rd week, where the part where diencephalon will be there, where the diencephalon part of forebrain will be there. Here there is an outpouching of the neural tube. So this outpouching is called optic groove. As this groove grows, it becomes bubble-like, it becomes vesicle-like. This will become optic vesicle, and as the vesicle enlarges, it goes and it approximates the surface ectoderm above that area. So as it does so, this optic vesicle expands and the connection with the diencephalon starts to narrow down, and this will become optic stalk, and this vesicle remains here. We will discuss the optic cup in the next slide. Let's see what is happening on the surface. As the optic vesicle goes and touches the surface or approximate the surface, it induces the surface to make a thick layer of ectoderm, and specialized layer of cells are generated here. These becomes the thick layer is called lens placode. This is going to make the lens in future, and then it starts both the lens placode and the optic vesicle begins to dip in the interior, or they begin to invaginate. So this is invaginating lens placode. This will become lens pit. So here is the part in the embryo where the lens placode is forming. Roughly day 22 to 24 will be this embryo age, and here these are picture from Langman's embryology, and here this is shown, the lens pit is shown, and this is the lens vesicle or lens that is developing lens vesicle, and this is the optic vesicle that is now folded upon itself, so making the optic cup. So let us see this process. As this invaginates, this optic vesicle, it folds upon itself to form the optic cup. So the part that is facing the lens will become the inner layer of the optic cup, and that part that is facing outward will form the outer layer of the optic cup. This usually starts just a few days after that, that is about day 22, and this space that remains between them, it is the continuation of the cavity of the neural tube. This is called the interretinal space. So here is the picture again. All these pictures are from Langman. So this is now the lumen of the neural tube that is extended into as the lumen of the optic stalk as the lumen of the interretinal space. So as it develops, this becomes the outer layer and this becomes the inner layer of optic cup, and between them is the interretinal space. And in the following weeks till the 7th week, this layer grows enough and they become approximated, obliterating the interretinal space between them. I have used the word these two layers fused, but this fusion is not so much anatomical, rather functional, and this remains a weaker point in our retina in the case of any high blood pressure, hypertension, or trauma, or many other causes. This may separate, so this remains a potential point. Optic cup does not only form by invagination of the cup here, but it also invaginates on the inferior or ventral aspect. So this fissure, this opening created by the folding from the inferior aspect, this is called choroid fissure. This allows the mesenchyme to enter into the cavity between the optic cup, this area, and this also allows the hyaloid artery, we'll see that shortly, it enters into the area to supply the structures inside. As the time passes till the 7th week, this part, this ring that is left behind, this opening will be the future pupil from where the light enters. So these are again back those pictures we saw in the previous slide. These are showing the lens pit, this lens vesicle, this is the inner layer of retina, this is the outer layer of retina. As you can see in this picture, the interretinal space has been obliterated. Again, a little more advanced form where the lens pit has disappeared and lens vesicle has separated. So here is the lens vesicle from the surface ectoderm, inner layer of retina and outer layer of retina with the obliterated interretinal space between them. Let us now focus our let's focus on the lens. So we did that. As the vesicle approached the surface ectoderm thickened to form lens placode. As it invaginated, it becomes invaginated lens placode that became lens pit, and as it goes further, it pinches off as lens vesicle. So this is the lens vesicle, a single layer of surface ectoderm pinched off inside with the basement membrane on the outside and the epithelium in its apical side on this side. So this is the lens vesicle. So it's a simple layer, single layer of cells and a cavity between them. So it loses its contact within the 6th week, and as the time passes, the cells on the posterior aspect of the lens, they start to elongate and they start to approach the anterior surface, and as they approach, they ultimately reach the anterior surface by roughly 7th week. These are pictures from Larson's Embryology. So this is the posterior surface, the anterior surface. As they elongate, they reach the anterior surface, obliterating the cavity, and this obliteration is complete by 7th week. And as they do so, these cells, all these cells of the posterior layer, they lose their nuclei and they get filled by crystalline, and these fibers are called primary lens fibers. So the posterior epithelial cell, they disappear by conversion into the primary lens fiber. The anterior epithelial layer remain behind the basement membrane, that is now called capsule, and they keep multiplying and going and keep being pushed to the periphery, to the equator, and at this point, at the equator point, they start elongation and they expand in this direction. So throughout the life, they are being added circumferentially on the outside from the equator part, and these lens fibers that are added, these are the secondary or cortical lens fibers. So the fibers generated in your childhood will be near the center. The fibers generated with the advancing age will be more and more toward the periphery, and these cells also lose their nuclei and they are also filled with crystalline molecules. So here is a picture video that shows the images. So this is the development of neural fold. So this is the neuroectoderm, neural crest, and surface ectoderm. Up at the point where the for future diencephalon will be formed is the eye field, and initially there's a common eye field, and gradually with the foldings they go separately. And these eye field, this is the part where optic groove will be created, and this optic groove, as it grows, it will become optic vesicle. As it approaches, continues to grow and approach the surface, it gives inductive signals to the skin and to the surface, and these inductive signals will lead to the changes in the skin and the changes in the reciprocal induction, and these will lead to invagination of both the skin lens placode and the optic cup. And as this is folding, this will become lens pit and lens vesicle, and these will be differentiated into two layers of retina. The inner one will be the inner layer of retina, the outer one will be the outer layer of retina. This will become neural retina, this will become pigmented retina, and in between them there is interretinal space that will be gradually obliterated. So before moving on to the further lectures, you may pause the videos as I'm going to discuss a few MCQs. So the optic stalk is developed from neuroectoderm, surface ectoderm, or mesenchyme. As it is coming from the neural tube, it is developing from neuroectoderm. The retina develops from, again, it is developing from diencephalon, then optic cup, so it is developing from neuroectoderm. So lens fiber that are produced by elongation of cells in the posterior face. So these one, are we talking about primary fibers or secondary? Yes, primary fibers. Let us move on to the next part, that is retina, iris, and ciliary body. When all of this was happening, the optic groove, optic vesicle, optic cup, we ended up in this part where there is internal layer of retina and outer layer of retina. This internal layer of retina will ultimately form the neural layer, and the outer layer will form the pigmented layer. So this is neural layer of retina formed from the internal and pigmented from the external layer. So these are the two layers of retina that has to be formed, and this neural layer of retina will be further divided into two parts. The part that is actually photosensitive, that will be pars optica, and the layer of retina that we saw in the beginning of the lecture, that is going to cover the back of ciliary body and back of iris, those will be insensitive to light. So these will be pars ceca, pars optica, pars ceca. So let us see them here. So this whole part, there is the light sensitive, this is the pars optica, and this faint yellow line that you may be able to appreciate behind the ciliary body and the iris, so this is pars optica. Pars ceca, from pars ceca ciliaris and iridium. Okay, behind the ciliary muscles and behind the iris muscle. These are the pictures from Netter's atlas. So let's talk about the pars optica retinae. This part continues starts to multiplying rapidly, and these single layer of cell become pseudostratified, and then multiple layer of cells are formed, and they develop into at least 3 layers of cells. The cell that are bordering the pigmented layer most away from the vitreous chamber, so there will be the rods and cones layer. Photoreceptive layers will be developed here. Here the rods, the cells that are sensitive to detect color, so that are sensitive to detect black and white and light, and the cones that detect color. These will be developed in this part near the pigmented retina. They will have their receptors towards the pigmented retina and the cell bodies and axons away from the pigmented retina towards the chamber of the eye. And as you can see, the cones, the rods are roughly 20 times more as compared to the cones. Then comes the central layer. This is the mental layer, and that contains the neurons that will contain the bipolar cells and the ganglion cell. We'll discuss it in this lecture too, and we'll go into further detail in the histology lectures. And also contains the supporting cells like Muller cells. And the topmost layer, or this is the marginal layer. Here is the exiting axons that gather towards the optic disc and exit through the optic nerve towards the eye, towards the thalamus, and then on the way to the telencephalon. So these are the 3 layers of neuron that are formed. At least you should remember these 3 cells most away from the cell center of the eyeball towards the pigmented retina. Somewhere here will be pigmented retina, so there will be layer of rods and cones. So rods and cones are technically the receptor part of these cells. So this is the layer of rods and cones, and then moving towards the center will be their cell bodies, their axons will be there. They will synapse with the dendrites of these bipolar cells. You can follow this image. And bipolar cells, they will give axon from the opposite end, and it will synapse with the dendrites of ganglion cells, and ganglion cells will have cell bodies, and they will give their axons as optic nerve fiber. So this was the mental layer. This was the layer of the optic nerve that runs on the interior surface of the retina on the way to the optic disc. So all of these layers gradually develop, and they are matured in this form by roughly 8th month of intrauterine life. We can discuss the detailed layer of structure of retina in the lecture of histology of eye available on the same channel. So just a reminder that light goes in this direction. Light starts from this layer, goes through all the layers of retina, and it's detected at this point. Then let us talk about the pars ceca retinae, the non-neural layer of retina. The two pigmented and non-pigmented layer, pigmented and neural layer, they continue, and both these layers continue on the inside of the ciliary body called pars ciliaris retinae, and they continue on the inner aspect of iris called pars iridica retinae. In the inside of the iris, and they cover the back of the iris. So this is the part of the iris that is covered with epithelium. This front part of the iris, this is made up of mesenchyme, that is mixture of development mixture of neural crest cell and mesoderm. And this mesenchyme give rise to fibroblast and melanocytes that make up the structures, the blood vessels here, the melanin pigment that gives you color of the eye. And this layer of epithelium on the back, it has pigmented layer on the anterior aspect that is very important for making this iris translucent to the eye, so that image light can go only from the pupils. And also this epithelium, this also gives rise to the muscles of the iris, that are the dilator pupillary muscles and the sphincter pupillary muscles. So if anyone asks you what is the origin of these muscles, so these muscles will be ectodermal in origin, because ectoderm was making neural tube, diencephalon, then optic vesicle and optic cup, and inner layer of retina and outer layer of retina. So these are going to make the muscles of the iris. So here is the mature form shown. Then there is ciliary body. Ciliary body is the one that is responsible for hanging the lens, and that is also responsible for generating the aqueous humor. So these are the numerous folds. These are called ciliary processes, and the epithelium covering this fold are the epithelium of the pars ciliaris retinae, the parts of pars ceca retina, and these epithelial folds serve two embryological functions. This is the epithelium that gives rise to the suspensory ligaments that hold the lens. Also, during the adult life, this is the epithelium that exudates the fluid of the aqueous chamber. So these are the epithelium, and on the inside there is mesenchyme. Again, the mesenchyme is of the mixture of mesoderm and neural crest cells, and these muscles, as opposed to the iridial muscles, they are generated from the mesenchyme. So let us quickly revise. The posterior 5/5 of the optic curve develops into pars ceca or pars optica retinae. It develops into pars optica retinae. The pupillary sphincter and dilator muscles are developed from, these one are being developed from neuroectoderm. The muscles of the ciliary body are developed from, these are being developed from the mesenchyme. You will have a little conflicting statement from different books. Some books will say mesoderm and some book will say neural crest cell. So mesenchyme is a safe answer to give, as those are working together to give rise to these muscles. The epithelium of the ciliary processes is developed from, again, neuroectoderm, because it is a part of the optic cup. Optic nerve, where does that come from? So as the retina develops, the axons of the ganglion layer of retina, they extend on the surface and they exit the retina through optic cup, optic disc, and they extend into the optic stalk, and that becomes populated to make the optic nerve. The optic stalk had the similar inner and outer layers. As it was folding, choroid fissure also made it fold, so we will have inner layer of optic cup, optic stalk, and we will have outer layer of optic stalk, and we will have the lumen of the optic stalk, just as was in the retina. So don't get it confused that this layer is not making the axons. The axons are coming from the retina, traversing through this layer, the lumen of this layer, the inner layer of optic stalk. And as more and more axons come, this layer will become more and more thick until in about 8th week this lumen is obliterated. But don't think that this is useless. This inner layer of optic stalk gives rise to the supporting cells, the astrocytes, that are going to support the cell bodies and axons there. And the oligodendrocytes, some says that they develop here, but some says that they migrate from optic chiasm and they move on to the eye till afterwards. So this myelination, it starts in the fetal period in the last few months, and it goes on until it is completed by the 3rd month, by 10th week of the human life. Here is one more thing about the halide artery. As the choroid fissure is formed, it gives place for the halide artery to enter the optic stalk, and with time, by the 7th week, when choroid fissure is closed, the halide artery is trapped in the center as central artery of retina. By the way, I talked about the optic nerve being covered by oligodendrocyte and astrocyte. Those are the features of the central nervous system, and that is why embryologically and histologically speaking, optic nerve is considered a part of central nervous system, because it is being myelinated like central nervous system by oligodendrocyte. It is supported by like central nervous system by astrocyte, and it is surrounded like central nervous system with the 3 layer of meninges and dura, 3 layer of meninges and the CSF. So the although it is counted as the peripheral nerve, but it is part of central nervous system embryologically, and that's why this is the only nerve that is affected by the demyelinating disease of CNS. Okay, now we are left with the outer layers, the choroid, the sclera, and the cornea. During the early weeks where all of this was happening, where optic curve was being formed during the 4th and 5th week, the surrounding area of the optic curve was surrounded by the mesenchyme. So this is completely surrounded by mesenchyme by the 5th week, and this is made up of neural crest cell and the mesoderm. This mesenchyme, it differentiates into 2 layers. The inner layer that will develop into a more vascular layer that will form the uveal layer, choroid, ciliary body, and iris. So this is the layer. This layer is embryologically continuous with the pia matter and arachnoid matter of the meninges, that is the leptomeninges. And then there will be outer layer that will be more fibrous in nature. There it will be less vascularized, and it will form the sclera and cornea on the outside, and this will be continuous with the dura matter. So as it is surrounding it, this condensation of mesenchyme in front of the lens and behind the surface ectoderm, so this area starts to split by generating small vacuoles. As the small vacuoles generate, they coalesce with one another and they coalesce to form a large chamber. This is called the anterior chamber. As this is broken up, so this layer of the condensation of mesenchyme, this is split into two parts. The part that is near the surface, so this is the part that will form the cornea. To be more specific, it will be forming the stroma of the cornea. Moreover, this dotted line, this will be formed, that will be formed the lining on the inner aspect of cornea, that will be the endothelial lining of cornea. There is some debate there. Some book says that Larson says that the stroma is made of neural crest cells, and this dotted line, the endothelium, is made up of the mesodermal cell. But again, this is a part of debate, and you will see different statement in different books. So this, after the splitting, this is the outer layer that will form the stroma of sclera, and this endothelium of the sclera. You can safely say that both of these are formed by mesenchyme, and if you want to take a risk, you can say that this is formed from the mesoderm and neurocrest cell, the stroma, and mesoderm forms the endothelial layer. And then this is the part that forms the this is the inner split. So this is the part that forms the front part of the iris, and it will be continued in front of the lens too. So this membrane, while it is available, while it is present there in the eye, this is called iridopupillary membrane, or simply the pupillary membrane. So this part, this will form the front part of the iris, the part that was in front of the epithelium, that will be making the fibroblasts and the melanocytes, and this wall will cover the lens, but as we go on, this will be ultimately dissolved and you will get the open pupil. So this part, cornea, is ultimately made up of three embryological sources. As you can see, its outer surface is covered from ectoderm, surface ectoderm. As the eyelids make, this surface ectoderm continues to cover it. So its outer surface is covered by epithelium of surface ectoderm. Its major stroma is made up of mesenchyme, most probably of neural crest in the origin, and its inner aspect, this one, it is made up of simple squamous epithelium. So this will be endothelium. This is again made up of mesenchyme, most probably of mesodermal in origin. Again, there is a conflict. The Keith Elmore says it's both neural crest, but Larson says it's mesodermal in origin. Then there is posterior chamber, the part behind the iris in front of the lens. This is the posterior chamber, and it is simply made as this vacuolation continues, and iridopupillary membrane is dissolved and it disappears. And when it disappears, posterior chamber and anterior chamber, they become freely communicating with one another, giving pathway for the aqueous humor to go from the posterior chamber to anterior chamber. We'll again discuss in detail in histology how it's produced from here, goes through the pupillary opening and to this angle, iridopupillary iridocorneal junction, and here the trabecular meshwork and the canal of Schlemm gives the place for drainage. Then we're left with vitreous body, the central part. So this whole empty place of retina in the optic cup, this was called the lentiretinal space. Lenti for lens, retina for retinal for retina. This space is invaded by mesenchyme containing both the neural crest and mesodermal cells, and the cells from there make up the primitive vitreous humor. As the maturation goes on, this inner layer of optic cup also helps in the production of vitreous body. This is a gel-like substance and it's covered by a gel-like membrane. So there will be a membrane outside and gel inside. In the earlier stages of development, there was a halide artery going from back to around the lens, forming this network of vessels around the lens called tunica vasculosa lentis. But with time, this artery gradually disappear, as does this capillary network, and in adults you are left with this canal where the hyaloid artery was there. So this was called hyaloid canal. So these are pictures from Keith Elmore's Developing Human. They have shown the same steps in little detail. You can pause the video here and go through these pictures if you want. So let's summarize. Where does the eye develop from? The neuroectoderm was responsible for making the two layers of retina, pigmented and neural, and neural later for further divided to optica and ceca was further divided to iris and ciliary parts. So it makes the posterior layer of iris, it makes the posterior layer of ciliary process, and from there suspensory ligaments, and through retina it also makes the optic nerve, and through this layer of iris it makes the pupillary muscles. Surface ectoderm is responsible for making the lens, the outer surface of cornea, corneal epithelium. We will see in the following slides the conjunctiva and lacrimal glands. Mesoderm and neurocrest cell, you can combine both these headings under mesenchyme, but I'm quoting Larson's embryology here. So mesoderm is responsible, both are responsible for making choroid and scleral layer. According to Larson, embryology in the mesoderm is responsible for making corneal endothelium and angioblast of choroidal layer, and neurocrest responsible for making corneal stroma, ciliary muscles, fibrous sclera, and vascular choroidal layer. We are left with the structures that are outside the eye now. These are eyelids. These are formed by these ridges, these prominences that are developed in the 5th to 6th week. The one on the top will be the, you can simply say superior or inferior, or this is called frontonasal and this is called nasal extension. So they start to grow, and as they grow and continue towards one another, you can see the surface ectoderm that was present between them starts to get trapped between the two advancing processes, and this whole part, this is forming the conjunctiva, the layer of epithelium that is covering the front of the lens and back of the eyelid, that allows for the minimum friction. This is simple layer of cuboid, this is stratified cuboid columnar epithelium with a lot of goblet cell that allows minimum friction. So these part where the folds upon itself, these are the conjunctival sac, and this forms the epithelium of cornea too. So embryologically this is one continuous layer, and as they approach one another they join and fuse by the 8th week of intrauterine life, and they remain fused all the way till the last trimester, where they get separated in 26 to 28 weeks, and some books says up to even 7th month to 8th month. So it gets separated pretty late in embryonic life, and you can see this if you have seen the pictures of newborn rats or newborn rabbits. They are born before this step and they have their eyelids fused. Accessory structures, the how the eyelashes are formed, how the tarsal plate, so eyelashes and glands, they follow the pattern of the cutaneous. For this, I will refer you to the lectures of the embryology of skin, where we'll discuss how the hair follicles are developed, how the glands are developed. So it follows the same pattern of the glands. The tarsal plate is the fibrous plate inside the eyelid on which the muscles is attached. So this is developed from the mesenchyme that was inside it. Orbicularis oculi muscle that is present inside the eyelid, that is developed from the mesenchyme of the second pharyngeal arch, and also supported by the muscles of that nerve of that arch too, that is the facial nerve, and the lacrimal gland that is developed from the outgrowth of the conjunctival sac. So lacrimal gland develops from here. So as it was an extension of the surface ectoderm, so any gland that is formed by this also will be developed from the surface ectoderm. So the lacrimal gland, although it does develop intrauterine, but it becomes functional about in the 2nd month of life. So it becomes functional by 6th week. So if you may have noticed that newborns don't produce tears, they start producing tears when crying in the 2nd month of their life. So that's about the embryology. Let us correlate with some abnormalities. And there is coloboma. This is keyhole defect where the iris is deficient. Like here or here, it is due to the imperfect closure of the choroid fissure, and it is due to the insult Any insult may be due to rubella virus, due to something else before the closure time, that is before the 7th week of intrauterine life. Iridopupillary membrane may be persistent and it may be covering the pupil. It's usually asymptomatic and light just passes through them fine, but if it is creating any problem, this can be removed surgically. Synophthalmia and cyclopia. Synophthalmia means fused eyeballs and cyclopia means single eyeball, and this is a midline defect. You must have studied this in the initial chapters of embryology or in general embryology or in the neural development of nervous system. So when there is issues of sonic hedgehog and a vast cascade of other proteins that are responsible for splitting on the right and left side, sometimes this does not occur very nicely, and the midline defects will be there, and this midline part is fused, on the halves are fused, and the brain will not have the two lateral ventricles. It will have a common central ventricle called holoprosencephaly. It will not have two prominences making the either side of the nose. It will have proboscis in the center, and you can follow them down. Aniridia, absence of iris, congenital cataract, presence of opacity in the lens, and congenitally it is major causes rubella infection. There are a lot of other causes including mother's diabetes, hyperglycemia, and a large number of infections too. Microphthalmia, small eyeball, and it occurs if the abnormality occurs before the 7th week, before the choroid fissuris closes, then the abnormalities will be more prominent because the optic cup was not developed. If the abnormalities and insults occur after that, then eyeball is usually symmetrical but smaller in size. If eyeball is not formed, then the orbital socket also does not form very nicely. So anophthalmia means absence of eyeball. Congenital aphakia means congenital absence of lens. Congenital ptosis means drooping of eyelids. It may be due to a number of causes, due to development of muscles, or the sympathetic splay effect will be there, or something else. So thank you so much for listening. If you have further questions, suggestion or feedback, please do mention in the comments below, or you can join us for any of our social media handles. Till next section, till then, Allah Hafiz.